This conference will now be recorded.
Students, I'm Dr. Divya from TeenEinstein. So in the 10th chapter, we are learning about microbes in human welfare. So in this, first we were discussing about the role of microbes, and overall introduction we have learned like how these microbes are involved in our daily life, or they are presenting the role of microbes in the soil, air, water, and some are present in our body also. So in animals, plants, and all, in their body itself, it is present for different functions. So when where the normal life is impossible, there we can see these microbes are living. Like extreme hot and cold conditions, these microbes will be present, and different types of microbes are there, like uh, protozoa, plant virus, animal virus, bacteria, fungi, all these are coming under microbes. So some more types are there, like we learned about prions and viroids and all. So all these are coming under microbes. Okay. Then in different roles, we can see this microbes in household products, how they are involved in household products. We have learned about that, about lactic acid bacteria we learned, and how that batter fermentation of that. We learned about Saccharomyces cerevisiae, we learned about that microorganism. Then in industrial products, what is the role of microbes in industrial product? We learned about fermenters, right? Fermentation of beverages, fermented beverages we learned. Then we learned about antibiotics. It's from microbes. Then chemicals, then enzymes, then bioactive molecules, all these things we learned. So next we are going to learn about uh, microbes in sewage treatment. Okay, in sewage treatment. So wastewater, we know about the wastewater and how it is getting treated. So every day in towns and all, wastewater amount, a huge amount or large quantities of that wastewater is getting produced town in town areas and all. So human waste or human excreta, that is a major waste in the waste material, right? So all these, how they are treating all this waste material, otherwise it may cause different kind of diseases, like so many pathogens may get developed from that. So wastewater, how it is getting disposed in the cities and all. See, we can't pour that into the natural water bodies, because that may cause different kinds of diseases, because that water we are using for drinking purpose, and other normal people are using that water. So we can't discharge that waste material into natural water bodies. So what we will do, so before disposal, see that sewage is treated in sewage treatment plants. So where they will treat that? In the sewage treatment plants, in short form STP, sewage treatment plant. So where we will treat these waste or discharge of this, or the wastewater which is coming in, which is getting created in the cities or in large quantities. Sewage treatment plants will treat that waste. So before disposal, the sewage is treated in sewage treatment plant, and then it is getting released. So what will happen, instead of creating such a pollution, the percentage of pollution, that will be very less. Up to that level, that will get treated, okay, the sewage. Then after that, that what they will do after that treatment, it will get discharged. So the percentage of pollution will be less. So the treatment of wastewater, how it is done? The treatment of wastewater is done with the help of heterotrophic microbes which are naturally present. Okay, which microbes are naturally present in that sewage. With the help of that microbes, the heterotrophic microbes, this treatment is done, and that treatment, sewage treatment, is taking place in two steps. Okay, first is the primary treatment and second step is the secondary treatment. Okay, first primary treatment, then secondary treatment. So the sewage treatment is taking place in two steps. First one is the primary treatment and second one is the secondary treatment. Got it? Primary treatment and secondary treatment. So in the primary treatment, first what they will do, it is the physical removal of particles. Okay, physically the large particles which are present, large particle in the sense the floating particles and all will get removed, and then it is getting filtered. First what they will do, this is the physical removal, the primary step or primary treatment, or the first step is the primary treatment. In that, that is the physical removal of particle. That means large and small particles which are getting removed through this primary treatment. Okay, so first what they will do, the primary treatment will be done, which is the physical removal of this large and small particles which can be filtered or which can be removed. Okay, so what they will do, it is done in different stages. First what they will do, the floating debris that is removed by that will get removed, the floating debris. First the floating particles will get removed, then it is removed by sequential filtration. The floating debris is removed by sequential filtration. Through sequential filtration they will remove the floating debris, then the grit are removed by sedimentation. What do you mean by grit? Soil and small pebbles. The small pebbles and the soil particles will get removed. That this is a sequential process. Okay. So the primary sludge that will get formed, that means all the waste particles which is settled, that solid that settle, that forms the primary sludge, and the supernatant, the after removing all this, in the primary stage, after removing all this through filtration, the supernatant forms, that is the effluent. The supernatant that which is formed, that is the effluent. So that effluent from the primary settling tank, it is moving to the secondary section, or the secondary, it is moving for the secondary treatment. From the primary section or the prime, after the primary treatment, that effluent will move to, so in this step what will happen, after primary treatment, that effluent is moving for the secondary treatment. Okay, for the secondary treatment, that effluent is moving. So that's all about primary treatment. So first, through filtration, that is a physical removal of particles, large and small particles will get removed through filtration and sedimentation. What and all they will do? Filtration and sedimentation. See, if you're filtering that, the large particles, first that large floating particles will get removed, then sedimentation, the all soil particles and all will get sedimented through sedimentation also. So these processes are going on for the primary treatment. Then after that, the remaining thing, that is called the effluent. After the primary process or the primary treatment process, the effluent will move towards the secondary treatment process. Okay, then in this, that primary effluent, then it is passed into large aeration tank where it is constantly agitated mechanically and the air is pumped into it. Okay, the second stage or the secondary treatment is otherwise called as biological treatment. Okay, secondary treatment is otherwise called as biological treatment. That means in the secondary stage or in the secondary treatment, it is getting treated biologically. So during that stage, that effluent is passing into large aeration tanks. Okay, that effluent after the primary treatment, that effluent which is formed, that will be passed into large aeration tank, and there it will get agitated constantly. It will get agitated, and after, and the air is also will be pumping into it. Okay, so this kind of treatment that will help in the growth of useful aerobic microbes. Okay, they'll form a group, that there will be a growth of aerobic microbes. Okay, microbes which will grow under aerobic condition, they will grow. So while growing, these microbes will use the major part of these particles or the organic matter in the effluent. For their growth, they need some nutrients, right? So this aeration of tanks and common constant agitation, mechanical agitation, uh, that will allow the growth of microorganisms. So microorganism, for their growth, for the growth of those microorganisms, they need some food material. So they will use the organic matter which is present in this effluent. They will use that organic matter for their nutrition, and that is used as a nutritive material or as a food material for this microorganism for their growth. Okay, so that reduces the that growth of that microorganism, that reduces the biochemical oxygen demand of the effluent. BOD means biochemical oxygen demand. Okay, so like that organic matter is used, it's being used by this microorganism. So that reduces the biochemical oxygen demand of that effluent. So this, what do you mean by BOD? BOD means amount of the oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by bacteria. Okay, if all the organic matter in 1 liter of water, it is being oxidized by bacteria, that time the amount of oxygen that would be consumed during the, in 1 liter of biological oxygen or biochemical oxygen demand means the amount of the oxygen, this is the definition you have to learn, the amount of the oxygen that would be consumed, the amount of the oxygen that would be consumed if all the organic matter in 1 liter water. Okay, in 1 liter water, all the organic matter were oxidized by bacteria. So that time, the amount of oxygen that would be consumed, that is called as biochemical oxygen demand. Okay, this is important definition, 1 mark question. And till the biochemical oxygen demand or till the BOD is reduced, up to that, this water, sewage of that water, uh, sorry, the sewage water treatment will continue. Okay, the treatment of that sewage water will continue till the biochemical oxygen demand is getting reduced. Or when it is low, up to that level, sewage water treatment will continue. Then in biochemical oxygen demand test, the rate of uptake of oxygen by that microorganism in a sample of water, and thus what is the biochemical oxygen demand test measures? The rate of uptake of oxygen. See, BOD test, if you are conducting, how or what it is measuring? The rate of uptake of oxygen by a microorganism in a sample of water. Okay, in a sample of water, the rate of uptake of oxygen by a microorganism that is getting tested. Okay, so what it is measuring, that biochemical oxygen demand, that means it is measuring the organic matter which is present in the water. Okay, organic matter, the amount of organic matter which is present in the water, that is getting measured by this BOD test. Okay, so when the BOD is greater, or the BOD of that wastewater is greater, the pollution or the polluting material which is present in that water is more. Okay, BOD more means it is having more polluting potential. Okay, BOD more means it is having more pollution potential. That is the meaning of that. Okay, then when that BOD or biochemical oxygen demand is getting reduced, when it is having a very low value, that means that is having very less pollution, or it is getting it is having very less pollution. Okay, so that time, that effluent, then it is passed then this effluent is passed into a settling tank. Okay, when the pollution level is very less, that time that effluent will get passed into a settling tank, and that bacterial flocks, they are allowed to sediment. Okay, when the BOD level is very less, or the wastewater BOD level it is very less, means that time that effluent will be sending to a settling tank, and that flocks, bacterial flocks are allowed to sediment. Okay, that sediment is called as activated sludge. So when it is getting sediment, that bacterial flock, when it is getting sediment, that sediment is called as activated sludge. Okay, then a small part of this activated sludge, it again, that small part of that activated sludge will get pumped into, or again it will be pumped back to that aeration tank. Why it is sending getting back to that aeration tank? Because that will serve as the inoculum. Okay, that remaining one, or which is pumping back to that aeration tank, that is working as the inoculum. Then the remaining part, after sending back that inoculum, the remaining part, that will be the major part, or more quantity, that will be more quantity. Then more quantity of that sludge, it is pumped into large tanks. Okay, that will be pumped into large tanks, and they are called as anaerobic sludge digesters. Anaerobic sludge digesters. That is called as anaerobic sludge digesters. Okay. Here, what is happening then? Again, some other kind of bacteria that will grow anaerobically, that microbes which is having the capacity to grow anaerobically, that digest the bacteria and the fungi in that sludge. Okay, then during that kind of digestion, the bacteria produces mixture of gases. That gas, while digesting, that bacteria produces mixture of gases. So what and all gases will be in there, in that mixture? Methane, hydrogen sulfide, and carbon dioxide. So in that mixture of gases, methane will be there, hydrogen sulfide will be there, and carbon dioxide will be there. So these combination of all these gases, that forms the biogas. Okay, so that is called as biogas, and this is working as an energy source which is inflammable. Okay, this gas is working, this biogas can be used as an energy source, and that is inflammable. Okay, then that effluent from that secondary treatment plant, that is, after all these treatment, that can be used, or that can be sent into, or that can be released into natural water bodies, because the level of pollution in that, or the pollutant will be very less. So that will be released into natural water bodies like rivers and streams. Okay, so this is the secondary treatment, or the biological treatment. This step is taking place with the help of or the microorganism, right? Some digestion of that material is taking place with the help of microorganism. And biogas, the production of biogas is taking place, which is inflammable, that is working as a source of energy. Okay, then after all this process, that effluent which is produced after the secondary treatment, that is released into natural water bodies. Natural water bodies in the sense streams and rivers. Okay, so that is the primary and secondary treatment of sewage treatment. Then see, now you know that how these microbes are playing very good role, or that their major role, okay, in wastewater treatment. So these practices are going on worldwide. They are taking these steps. So these are getting they are using these technologies in the treatment of sewage. Okay, then the Ministry of Environment and Forest, they have initiated Ganga Action Plan and Yamuna Action Plan. Okay, these are for saving the main rivers, for saving Ganga and Yamuna, for the major rivers of our country, major rivers of India from pollution. For saving Ganga and Yamuna, they have already started Ganga Action Plan and Yamuna Action Plan. Okay, this is the sewage treatment plant. The whole picture, you can see that a sewage treatment plant. Then microbes in the production of biogas. See, first now we learned about the in sewage treatment plant what is the role of microbe. Now we are going to learn about microbes in the production of biogas, how biogas is getting produced. So biogas, you know that it's a mixture of gases produced by the microbial activity, and this biogas can be used as a fuel also. So with the help of microbial activity, biogas is getting produced. Biogas means it's a mixture of gases, and this can be used as a fuel. So different types of gases are getting produced due to this microbial activities. So during the growth and metabolism, microbes are producing different types of gases, which are the end product of this growth and metabolism of so many microbes. Okay, so that type of gas which is produced, that is depending upon the microbe and the substrate they are utilizing. Okay, the type of gas produced as a product, that is depending upon which microbe is used and which organic material they are using for their work as a substrate. Okay, so for example, what and all we can say, fermentation of dough. In that dough, that is the substrate, and their microbes are getting involved. Then cheese making, then production of beverages. Then in all this, the main gas which is produced, that was carbon dioxide. Okay, then in certain bacteria, like anaerobic bacteria on celluloid material, that produce large amount of methane along with carbon dioxide and hydrogen, that will produce methane also, anaerobic or cellulosic or on cellulosic material which is having anaerobic actions. So on that bacteria will produce methane along with hydrogen and carbon dioxide. So those kind of bacteria are together called as methanogens, which is helping in the production of methane. They are called as methanogens, and among that, one common methanogen is methanobacterium. Okay, methanobacterium is one common methane producing bacterium. So such organisms, such bacteria, they are together called as, or they are collectively called as methanogens and In that, one common bacterium is methanobacterium. So methanobacterium, we know that this methanobacterium, they are commonly seen in anaerobic sludge during sewage treatment. So this methanobacterium, they are commonly seen during the sewage treatment, and they are at the same time they are present in the rumen. Rumen means part of stomach. In rumen also it is present. In the cattle, rumen also this is present. So you know that they are eating grass, hay, and also a lot of cellulosic material. Cellulosic materials, so much of cellulosic material is present in the cattle, or it is included in the food material of cattle, and that is present in the rumen. So in the rumen, what is happening? These bacteria are helping to break down the cellulosic material, and they are having a major role in the nutrition of that cattle. Okay, so the excreta or the dung of that cattle, mainly in cow we can say cow dung, so they are commonly called as gobar. That is very rich in these bacteria, and this dung can be used for the generation of biogas, commonly called as gobar gas. So with the help of cow dung and all, they can generate biogas. This with the if in one home cows there, means with the help of that dung, cow dung, they can create or they can generate the biogas that is commonly called as gobar gas. So let us discuss about a typical biogas plant. So in a biogas plant, you can see the picture of the biogas plant here. This is the biogas plant. In this case, a biogas plant that is having a concrete tank. Biogas plant is having a concrete tank, and in that bio wastes are getting collected. Okay, in that concrete plant, concrete tank, all these waste materials are getting collected, and a slurry of dung is fed. Okay, then there will be a floating cover that is placed on that slurry. So on that slurry, a cover floating cover will be placed. So when that gas or the gas is rising, along with that, this cover also will be raising. So when that gas is produced in the tank with the help of the microbial activity, what will happen? The gas production, when the microbial activity in that slurry it is getting increased, along with that gaseous production also will be more. So along with the gas level, this also will keep on raising, right? Then that biogas plant will be having one outlet. That biogas plant, it is having one outlet. So that outlet will be connected to a pipe, and through that pipe, that biogas will be going outside. That biogas which is formed inside this will be moving outside. Then that spent slurry, that will be removed through another outlet. So through another outlet, that spent slurry will be removed, and that will be used as a fertilizer. And that can be used as a fertilizer. And for what and all purpose we are using this? For the like this produced biogas, for cooking and lighting purpose we are using this biogas. Okay, then So that's all about the biogas plant. We have learned about the structure, how it will be that biogas plant and how it is getting released after the production. Next, we are going to learn about microbes as biocontrol agents. Okay, microbes as biocontrol agents. That's what we are going to learn now. As biocontrol agents, how these microbes are working. So what do you mean by biocontrol? Biocontrol means for controlling the plant disease and plant pest, we are using this biological method. So that is the biocontrol method. Biocontrol means the use of biological methods for controlling plant disease and plant pest. Okay, so what do you mean by biocontrol? Use of biological method for controlling plant disease and plant pest. So that is what we are doing with the help of chemicals. Some spraying and all will be done so that we can kill this disease causing agents and all. So by the use of insecticide and pesticide, nowadays we are controlling, in our society we are controlling this diseases or disease causing agents. But we know that all these chemicals, disease causing chemicals or the pesticides or these insecticides, they are very dangerous or they are extremely harmful. These insecticides and pesticides are very harmful to human beings, not only for human beings, for animals, all those plants, for everything it is, because they are chemicals. So they are getting or they are creating harmful effects in animals, human beings and all. And they are polluting the environment. They are creating water pollution, soil pollution and all. And they are polluting the environment. Along with that, soil is also getting polluted, right? So soil is also getting polluted through the use of what and all, some chemicals which we are spraying for weeds. That is, for weeds, what we will spray? Weedicide. Weedicide means the chemicals which is killing weeds. So weedicides which we are spraying. So such weedicides, for what purpose you are spraying? For the removal of weeds or to kill the weeds. So soil is also getting produced with the usage of this weedicides. So biocontrol means use of this biological method. All this biological method we need to use for the control of plant disease and plant pest. Okay, plant pests and plant disease should be controlled. For that, which method you are using, that is called as biocontrol. So normally with the help of chemicals we are controlling. In the society we are controlling with the help of pesticide and insecticide. So these and all these pesticide and insecticide, all these are harmful or it is creating dangerous diseases. So along with that, it is polluting the whole environment, like water pollution, soil pollution, everything is it is creating. And soil pollution or soil, how it is getting polluted? Through this weedicides, for the through the usage of this kind of chemical sprays, weedicides and all. Okay, so how to control this biologically, these pests and diseases? How to control this? So instead of introducing these chemicals, the biological control method, how it will be, or what they are doing in the biological control method, that natural predation, natural predation. Okay, so instead of chemicals, they are using natural predation. So generally what we are thinking, biodiversity that increases the health or biodiversity furthers health. So the more variety a landscape has, the more sustainable it is. Okay. If a landscape is having more variety, it will become more sustainable. So for that, what they will do, that the farmers need to do what they should do. They have to maintain or manage the level. Instead of killing the whole organism, they should manage the level of these organisms to keep the balance with that within that ecosystem. So they have to manage the level of that ecosystem, because ecosystem, it is very complex. So instead of killing or instead of eradicating all these pests, they have to manage the level within the system. They have to manage. Okay. So contrary to the conventional farming practices, that is, in that farming practices, the chemical method to kill all the organism, that is very harmful actually. So what we need to do, we need to develop an understanding of the web. That means we should manage the level of all organism, and that interaction between the organism, that should be managed, or that level should be maintained. So this mutual controlling of this organism, or this biocontrol measures, the measures which are taking for the biocontrol, that helps in the usage of, or that helps to stop the usage or to reduce the usage of this sprays and pesticides and other chemicals. Okay. So the biological farming, in that, most important thing is, we need to be familiar, we need to have a clear idea, or we need to be familiar with the different forms of life, or different life forms which is present in the field. Okay. And we should have a clear idea about their life cycle. So we should know about their life cycle, their feeding pattern, and the habitats, and how they'll prefer or how they will live. So that all these, if we are knowing about one organism completely, we are knowing, we have a knowledge, a clear idea about one organism, the life cycle of organism, when it is getting that reproduction, or after reproducing, how it is growing and how it is getting multiplied. If we are having complete idea about one organism, we can take the control measures, right? So that time, that biocontrol measures we can take, so that won't affect completely the production. Red and black markings in one beetle, that is a common beetle, the ladybird. And here, this is the ladybird, and this is the dragonfly. These are helping to get rid of aphids and mosquitoes. Okay, the presence of dragonfly and this ladybird, this will help to reduce aphids and mosquitoes. So for an example, we can say the microbial biocontrol agent that can be introduced in order to control butterfly caterpillar, that is one bacteria, Bacillus thuringiensis, bt. Okay, this has created a lot of issues, bt. So microbial control agents which can be introduced. For that, one example is Bacillus thuringiensis, which can control butterfly caterpillars in butterfly caterpillars. Which bacteria? Bacillus thuringiensis. What is the name of that bacteria? Bt, Bacillus thuringiensis, which is introduced to control butterfly caterpillar. But these Bacillus thuringiensis that can be available in sachets, and how it will be? It's like dried spore, and these are mixed with water and sprayed onto plants. Okay, how it is used? These are mixed with water and that can be sprayed on plants and fruit trees. On fruit trees, you can spray this. Then brassica, you can spray on brassica and fruit trees and all. And this insect larva, they will eat this. Okay, when that insect larva is eating this, in the gut of that larva, the toxin will get released. Okay, so in the gut of that larva, this toxin is getting released and that larva will get killed. Okay, that time that larva will die. In the gut of that larva, this chemical, this toxin, it will get released and that larva will die. Then that bacterial disease that will kill the caterpillars, but other insects, this won't infect, or this won't affect other insects. Okay, other insects will get protected or they'll remain unharmed. So genetic engineering, they developed this kind of methodologies or methods. Then with these findings, that scientists, they introduced Bacillus thuringiensis toxin genes into plants. So Bacillus thuringiensis toxin genes scientists introduced into plants. And that plants, when it is getting introduced, Bacillus thuringiensis genes are getting introduced into plants, those plants will become resistant to this kind of insect pest. Okay, so in those plants, this kind of insect pest won't work out. For example, bt cotton, Bacillus thuringiensis cotton, in that plant itself, that toxin, that gene, bacillus, they have introduced Bacillus thuringiensis toxin gene in that plant. That means bt cotton, in bt cotton they have introduced Bacillus thuringiensis toxin genes. And this bt cotton is cultivated in some states in India. So Bacillus thuringiensis, how it will work? These are available in sachets, and how it is present as dried spores. This we can mix with water and we can spray onto the plant. Okay, the insect larva will eat this, and in the gut of that larva, that toxin will get released and that larva will get killed. So that bacterial disease that will kill this bacteria, that will kill this caterpillar, not bacteria, that bacterial disease will kill this caterpillar, but they won't leave, they won't affect other insects. Okay, there won't be any harmful effect for other insects. And that's how this genetic engineering developed this kind of methods and all, and they introduced this Bacillus thuringiensis toxic gene into plants. So those plants are resistant to the attack of these pests, insect pest. For example, bt cotton. So in bt cotton, they have introduced such genes, and such this bt cotton, it is cultivated in different states of our country. Okay. Next, this kind of biological disease or biological control, it is so that biological disease control or that biological control, it is developed for the plant treatment or plant disease treatment. So like that one is trichoderma, fungus trichoderma. Okay. So you know about this trichoderma. Trichoderma means it's a free living fungi, and that is very common in the root system. Okay, this trichoderma, fungus trichoderma, it is free living fungi and it is very common in the root ecosystem. So they are working as a best or effective biocontrol agents for several pathogens. So for several pathogens, they are this trichoderma, it is working as a biocontrol agent. Then baculoviruses, they are the pathogens and that can attack insect and other arthropods. So in that, mainly or majority of these baculoviruses, they are used as biological control agent, and they are in the genus nucleopolyhedrovirus. Nucleopolyhedrovirus. Okay. So these viruses, they are excellent candidates for specific species specific narrow spectrum insecticidal applications. So narrow spectrum insecticidal applications, these viruses are very excellent for that species specific narrow spectrum insecticidal applications, and they won't create any negative impact on that plant, mammals, fish, and which insect is not targeted. For such insect also, they won't create any issues. So this is most desirable when that beneficial insects also will be there, right? So when those insects are getting conserved, or if for those insects nothing is happening, that that will create positive sides, right? So this is especially desirable when beneficial insects are being conserved to aid in an overall integrated pest management program, IPM, overall integrated pest management program, or when all these are ecologically sensitive areas being treated. When an ecologically sensitive area is being treated, that time it will become desirable when beneficial insects are being conserved. When through all these things we can conserve beneficial insects, and or otherwise we can say ecologically sensitive areas, it is being treated. Okay. Next, microbes as biofertilizers. We are going to learn microbes as biofertilizers. Okay. So hope up to this everything is clear. Next topic is microbes as biofertilizers. In this we have to learn so many things. So that we will learn in the next class. So up to this, do you have any doubt? Is it clear? You want me to repeat any topic? Is it clear? I hope this is clear. So in this topic, you have to learn about Bacillus thuringiensis, or the biological control of pests and diseases, which and all we are controlling, and how this Bacillus thuringiensis is working. So that is an important thing. Then after that,